So the next topic that we see here in chapter 3 is cell division. Cell division is the process by which certain cells will divide themselves into two daughter cells. And so what we see here on this page, this is the cell cycle. So that's going to be the other thing that we're going to talk about here on this page here. So the cell cycle refers to the life cycle of the cell, and there are a few subphases within this, the cell cycle that we're going to note. So we have interphase, we have mitosis, and we have cytokinesis. So while this is a cycle, and the cell cycle doesn't really begin in one particular spot, I like to imagine that it's going to begin right here at interphase. So interphase is going to be made up of three other phases. We have the G1 phase, we have the S phase, and the G2 phase. So the G1, S, and G2 phases all are subphases within interphase. Mitosis is going to be made up of prophase, metaphase, anaphase, and telophase. Those are going to be the four phases of mitosis, and then telophase is going to be followed up with cytokinesis. Cytokinesis just refers to another phase in here. It's actually not shown on this picture, but it exists right in here after telophase. So this is where cytokinesis is going to occur. It's going to go right after telophase. So you'll often hear telophase and cytokinesis together as they are sort of happening at the same time. They're happening very close together. So cytokinesis is going to be that third phase that happens right here between mitosis and starting interphase again. Now we also have this G0 phase. The G0 phase is what is known as cell cycle arrest. So cells are going to enter this G0 phase and sort of move their way away from the cell cycle, and they're not going to grow anymore. But let's talk about each of these phases. Break down the individual components of each phase. So interphase, I like to think about as our cell's adolescence. This is the adolescence phase of our cell cycle. So the G1 and the G2 phases will characterize lots of cell growth and metabolic activity. We also have cellular organelles being replicated in these, but the most notable, in my opinion, the most notable subphase within interphase is the S phase. The S phase stands for DNA synthesis, and this is the phase where DNA is replicated. So while we have if we were to look at this picture here, through mitosis is the actual process of cell division. We take one cell and we're going to make two cells, and then each of these cells are going to undergo interphase. So these immature daughter cells are going to be progressing through interphase, and it's important that they are able to grow and have their metabolic activity. And in the S phase, their DNA is going to be replicated. So actually, in the G2 phase, as well as mitosis, we're going to have two copies of our cell's DNA. So every cell has 46 chromosomes. Every cell in the human body has 46 except for gametes. That means that in every S phase, those 46 chromosomes are going to be replicated. We're going to have two sets of 46 chromosomes, and they're going to be used whenever we divide our cell here in mitosis. So these are our three subphases within interphase, but what gets most of the spotlight here is the mitotic phase, so mitosis, so prophase, metaphase, anaphase, and telophase. So these are the phases where our cell is actually going to do the dividing. So we're going to talk about each of these phases as we go. First up is interphase. Now I've already mentioned that the interphase is made up of the G1, S, and G2 phases. And a few of the events, again, that occur in G1 are going to be cell growth, normal metabolic activity, preparing for cell division, replication of organelles, and so forth. There's a lot of things that happen in G1 and G2, but the S phase, I think, carries the most notable activity, and that is DNA replication or DNA synthesis. So we're creating this copy of our DNA. It's important that we have two versions so that we can pass on 46 chromosomes to both of our two daughter cells. So DNA replication is vital for our cells to actually be able to truly reproduce and create two identical cells. Now in interphase, our DNA or genetic material is going to be existing in the form of chromatin. So chromatin is going to be found in the nucleus, and we're going to have our nuclear membrane intact. That's going to be keeping all of our genetic contents inside the nucleus here. And it's important that we have chromatin because that makes DNA more accessible to the different factors that are going to come in and replicate that DNA. So as we look here at the replication of DNA, we're gonna note a few of the a few of the more prominent events that occur here with DNA replication. So DNA will always self-replicate. DNA replicates itself. That is going to be very, very unique to DNA. DNA is very special because it can take care of its own replication. And it does this with a few series of enzymes. Now, there are many, many enzymes, and if you ever take a genetic class, you will go over lots of the different proteins and other factors that are going to come in here and help for the replication of DNA. But for our class, I just want you to know two of the different enzymes and what their functions are. So, first and foremost, with the replication of DNA, it's important that our DNA becomes accessible. So we have to break these bonds in the center, we have to break these hydrogen bonds and separate the two DNA strands from each other. And this is going to be done with an enzyme known as helicase. Helicase is going to be an enzyme that's going to function to break the hydrogen bonds and uncoil DNA. This is going to allow DNA to be more accessible so that our next player can come in and add nucleotides. The next player is DNA polymerase. DNA polymerase's function, it's going to attach here onto the template strand. We'll refer to this as the template strand because it functions as a template while we are going to be forming this new strand of DNA. So DNA polymerase is going to come in here and it's going to add nucleotides according to the base pairs that it reads. So DNA polymerase is reading our DNA, it's reading the individual base pairs and giving the proper complementary base pairs as it goes. So there's also going to be a DNA polymerase over here also. So DNA polymerase adds the nucleotides while helicase is going to uncoil DNA. DNA polymerase is going to use the old strand as a template and it's going to form a complementary new strand. So DNA polymerase is adding these nucleotides as it goes and it reads the template so that it knows which is the proper complementary based pair. Now, there are a lot more enzymes and other factors that come in here to aid in the replication of DNA. Some enzymes are going to proofread. They're going to come in here and make sure that there are no errors that are occurring in our DNA and that we're copying everything properly. Some are going to fix DNA segments that have been damaged and so forth. So there's a lot of other factors that are playing a role in DNA replication. But as long as you know these two enzymes, those are the only two factors I'm going to want you to know about DNA replication. Understand that DNA is going to self-replicate and it's going to make a new strand out of the old strand. And that's going to give us two different versions, but we can call these copies because they contain the same information. They'll contain all of the same genetic information. They're just two versions of that DNA. So that's going to bring us into mitosis. Mitosis is the actual stage of the cell cycle where our cell is going to separate into two different daughter cells. Mitosis refers to nuclear division, or the division of the nucleus. So each nucleus is going to get a copy of each chromosome, and that's going to produce two identical cells. Now, our stages of mitosis are prophase, metaphase, anaphase, and telophase. PMAT. That's our mnemonic here. PMAT. This is the order in which our stages of the cell cycle are going to progress. So let's look at the cellular events that occur in each of these stages. Now, chromatin is going to be condensed into chromosomes. Chromatin, being its very large spaghetti structure, it's just going to sort of be a big ball of genetic material found in the nucleus. It's important that we can condense that chromatin and turn it into chromosomes so it can be packaged and processed easier. We can move condensed chromosomes around much easier than we can chromatin. So think about prophase and what's going to be happening here. We're preparing for cell division. So the nuclear membrane is going to be disappearing in prophase. It's important that this nuclear membrane disappears so that our cells and these chromosomes can sort of move around within the cell. They're going to be constricted by the nuclear membrane if it doesn't disappear. So this nuclear membrane is going to disappear. We start to have the formation of spindle fibers, and those are these little blue spindle fibers in here. We see the spindle fibers. The spindle fibers are going to be attaching to the kinetochore, the center of our chromosome, and they're going to be pulling on either direction. So this is going to happen in a later phase, but the spindle fibers are beginning to form here. And our centrosomes are going to be moving to either side. You can see that this is showing us early prophase, and this is late prophase. So it's important to remember that this is a progression of events. There's no time where these events just occur, every single time. This is a series of events, and these stages occur over time. So the centrosomes are going to move to either side of the cell, project these mitotic spindles, and prep our cell to be into the next stage, which is metaphase. So metaphase is characterized by the lining up of our chromosomes along the metaphase plate. I like to think that these are actually lined up along the middle of the cell, and middle begins with M, just like metaphase. So you can remember when you see one long line of chromosomes down the center or the middle of the cell, that's going to give us metaphase. But we could also call this the metaphase plate or the equator of our cell. So metaphase is very characteristic. You can see under the microscope, this is what it will look like here under the microscope. There's a long line of this genetic information that's going to be lining along the metaphase plate and is getting ready to pull, the cell's getting ready to pull these chromosomes apart. So we're going to get our first cell division here in just a second. So metaphase is characterized by this line of chromosomes that have lined up. Now we wouldn't have been able to have this if the nuclear membrane had not disappeared and we had not formed these spindle fibers. So all the events that occur in prophase are very important for allowing the events in metaphase to progress. That will lead us into anaphase. Anaphase is going to be characterized by the actual pulling of the sister chromatids apart and moving them towards opposite sides of the cell. So you can see in early anaphase, these spindle fibers are going to be pulling on these chromosomes, and that separates the sister chromatids from each other, and that's going to pull half of the genetic information to this side of the cell and half of the genetic information to this side of the cell. And in late anaphase, you can see over here, these are really going to be at two opposite sides of the cell. So anaphase is going to be characterized by having our chromatids pulled apart from one another and separating those chromosomes into two different sides of the cell. So this is nice. You can see early anaphase up here, so things are a little bit more difficult to see in this picture. But over here, you can see these nice two lines of DNA. And anaphase is characteristic because it's going to have two lines compared to metaphase, which will just have one. So anaphase is also nicely recognized here under the microscope. Telophase is our last phase. So in telophase, this is when our genetic information is going to be separated, and we're going to start forming the structures that we're going to need for two cells. So these mitotic spindles are no longer going to be needed. So they will dissolve. They're going to disappear. There's no longer going to be mitotic spindles. The chromosomes are going to become chromatin. They're going to decondense into that chromatin so that we can start growing our next cell. Again, we need to have chromatin so these different factors can access our DNA. It wouldn't be possible for any factors to access DNA if our DNA is condensed in the chromosome form. So our nuclear membrane is also going to form around each of these, and you can see we have two nuclear membranes as we are creating two different cells. But the other event that's very characteristic of telophase that you can always look for under the microscope is the formation of the cleavage furrow. The cleavage furrow is the location where the cell starts to pinch off, and this is going to cleave these two cells from each other. So the cleavage furrow, you can see here on the microscope, is another very characteristic structure of telophase. Now, cytokinesis is also happening at the same time that telophase is happening. Cytokinesis refers to the division of the cytoplasm. So, while mitosis referred to the division of the nucleus, cytokinesis refers to the division of the cytoplasm. So this is important that you understand the difference between mitosis and cytokinesis in this way. So cytokinesis is going to occur and happen at the same time that telophase is happening, and they're just referring to the respective parts of their cells that are being divided. So the cleavage furrow also is going to divide the cytoplasm as well, and it's also going to separate the two nuclei and form two new daughter cells. So we can see here that this cleavage furrow has been pinched off, and we now have two full cells. These are identical daughter cells, and they are now in interphase. They are now starting interphase. They're going to be growing and undergoing lots of metabolic activity, and soon they will start replicating their own DNA. So mitosis is the process that we just went over. But there's another process known as meiosis. So think back to all the times I've said that human cells have 46 chromosomes except for gametes. Gametes are the one cell in the human that are going to have 23. So meiosis is the process that only occurs in gametes, and this is going to help us to form new gamete cells with only 23 chromosomes. So when a sperm cell with 23 chromosomes meets with an oocyte with 23 chromosomes, that's going to form a new person with their own unique 46 chromosomes. So meiosis looks like it might be a lot more complicated, but all it is mitosis twice. That's all that meiosis is. So you can see that that with meiosis we're going to have prophase 1, metaphase 1, anaphase 1, and telophase 1. And instead of going back into interphase and growing and replicating our DNA, we're just going to immediately enter prophase 2. We're going to immediately enter meiosis 2, where we have the events of mitosis occurring one more time. So up here, we've replicated our genetic material, and we have 46 copied chromosomes. Through meiosis 1, those 46 copied chromosomes are going to be split between two cells, and those are going to be these two daughter cells. So each of these cells will now have 46 chromosomes, but those chromosomes will not be copied. Now, when we go through meiosis 2, this is going to be characterized by prophase 2, metaphase 2, anaphase 2, and telophase 2. And this is going to take our cells with 46 chromosomes that came from meiosis 1 and split them further so that we produce 4 gametes here with 23 chromosomes each. So these cells are haploid, which means that they just have 23 chromosomes. So that's all that meiosis is. Meiosis is just mitosis twice, and it occurs in our gametes. And there's some other things that go on here about meiosis. We could really break down meiosis a lot more than this, but this is what I will need you to know here for my class. So our final topic for Chapter 3 is going to be protein synthesis. Proteins do so much for our bodies, and so it's important that we understand how these proteins are made. So protein synthesis refers to the formation of proteins. Protein synthesis can be broken down into 2 stages. Those 2 stages are transcription and translation. Transcription is stage 1, and translation is stage 2. Now what we can see on this slide is that transcription is going to be occurring inside of our cell's nucleus. And then when we've synthesized some RNA from transcription, that's going to be the goal of transcription, is to synthesize RNA, that RNA is going to move out here into the cytosol. So what we see on the left side here is the series of events that is going to characterize protein synthesis. Protein synthesis is going to take DNA and turn it into RNA, and then RNA and turn it into proteins. So RNA is going to be what's actually creating the proteins, or serving as the template for our proteins to be created. And remember, we have a very important cellular organelle known as the ribosome that's going to be functioning in helping us synthesize these proteins. So with step 1 here, with transcription, that's going to characterize DNA going into RNA. DNA, we're going to form RNA from the DNA. So transcription here in the nucleus is going to take this DNA, make a strand of RNA. RNA is then going to leave the nucleus and go into the cytosol, where it's going to find ribosomes, and then we're going to take RNA and make a protein with it. So these are the events that are going to occur in protein synthesis. So let's break down transcription and translation individually. So transcription can be characterized as taking DNA and making some RNA out of it. So transcription more accurately refers to the formation of a strand of what is known as mRNA. That stands for messenger RNA, and that's this red strand right here. So you can see the purple strand is DNA, and we have our complementary base pairs and so forth. But what we also see here is that we have a new enzyme. We have another enzyme, and that is known as RNA polymerase. So this is very similar to DNA polymerase, but RNA polymerase is going to just add ribonucleotides and create a strand of RNA. So it functions in the same way that DNA polymerase does, but RNA polymerase is going to come in here and make RNA instead of DNA. So you can see that we're going to attach this onto our DNA, and it's going to read the template, and every time we have a guanine, we're going to add a cytosine. And every time we have a thymine, we're going to add an adenine. And every time we have an adenine, we're going to add a uracil, because remember, in RNA, we don't have thymine, we just have uracil. So you'll see, if you look at this strand here, all of the places that there were T's will now have U's. So uracil is going to be our nucleotide that can be found in RNA only. Now again, we could do a whole class on transcription alone, but I just want you to understand that we're forming this RNA out of DNA. That's the goal for transcription here. So we're transcribing our DNA code onto the mRNA. And the mRNA is then going to leave the nucleus, enter the cytosol, and attach to a ribosome where we can make new proteins. So translation is going to be the next step of protein synthesis. So with translation, we're going to be reading this strand of mRNA, and that's going to be telling us which amino acids we're going to link in which order to give us our protein. So you can see this polypeptide chain here. This is going to be forming a protein as we read our mRNA, and that's why our genetic code is so important, is because we got this code from the DNA. So the mRNA is just going to come in and take the code from DNA and then tell the ribosome which amino acids to put in here. So we're going to go over this translation here on the next slide also. But translation can more accurately be referred to as the taking RNA and turning it into protein. So again, protein synthesis takes DNA and eventually turns it into some protein. Transcription takes the DNA and turns it into RNA, and translation takes the RNA and is going to turn that into a protein. So this is a very big and involved slide, so make sure that you look at this in your books before you watch this associated portion. But before I get into this picture and talk about how these are going to work, I want to mention that there are more than one type of RNA. So mRNA is the type of RNA that was formed from our DNA. That's this red strand that we see here that contains our genetic code. But we also have something known as tRNA, or transfer RNA. And tRNA is shown here in this blue thing here. This is tRNA. tRNA, or transfer RNA, is going to have two major characteristics that we want to mention. And that is that every tRNA molecule is going to have an amino acid attached to it and something known as an anticodon that will complementary base pair with the codons that can be found on the messenger RNA. So the anticodon here are going to be different nucleotides, and based on the nucleotide that we have, that's going to determine which amino acid is going to come in and get linked next. So remember, when we talk about proteins are long chains of amino acids, and the different arrangements of those proteins, their primary structure, are going to build and set the stage for all of the other protein structures, so secondary, tertiary, and quaternary structures. So all of the protein structure is going to be very, very important. And so the individual amino acids are going to be determined by the anticodon that can be found on the transfer RNA. So in order to understand what an anticodon is here, we need to understand what a codon is, and that is on the messenger RNA. So we have codons found on our messenger RNA and anticodons found on the transfer RNA. Codons are simply a sequence of 3 nucleotides. That's all they are, just a sequence of 3. So you can see A, U, and G is going to be the start codon. Well, the next codon here is going to be U, G, and C. And so depending on which transfer RNA is going to have the proper anticodon to match with the codon, that's what determines which amino acid is going to come in here. So here we see the 20 amino acids. We need to get these 20, these are going to be the most common 20 that we find. We need to get these 20 to link up in some way, depending on our protein structure. Now, I don't want you to go and learn all the structures of each of these amino acids, but I want to show you that there are 20. And now that we can see the amino acids on this slide, I'll mention that the codon and the anticodon are going to match up, and that determines which amino acid is going to link to which other amino acid. So one example here, I'll just use histidine for an example. Histidine is one of the amino acids. When we go to this picture, this is the codon chart. So histidine is going to be coded for by the codon CAU and CAC. These codons are going to bring in a histidine. Now, I don't want you to focus on learning these different codons. I don't need you to know the different amino acids right now. What we want to understand is how this process of protein synthesis is going to work. So again, we have our messenger RNA, and that's going to bind to a ribosome. The messenger RNA is going to have a start codon, which is going to bring in our first transfer RNA molecule. And that is going to be determined by the individual sequence of the anticodon and the start codon. So the transfer RNA is going to bring in an amino acid attached to this transfer RNA molecule. The large subunit is going to come in here, and it's going to house our transfer RNA. And then a second transfer RNA molecule is going to come in here and be closely associated with the first one. And you can see that what's going to happen here is these are now found next to each other, and they're also all complementary base pairing with codon and anticodon. What eventually is going to happen is we're going to form a peptide bond. So these bonds are going to form, and that is what's going to form our protein. So remember, a protein is just a long chain chain of amino acids, a very, very long chain of amino acids. So we're forming these new polypeptides here as we go through. And you can see down here that as our ribosome moves along the mRNA, that's going to shoot amino acids out in a chain and eventually give us our protein. So this is going to function as an assembly line, and once this first transfer RNA molecule has given away its amino acid, this is going to expel itself from the ribosome, but eventually you can see that we're going to get this nice long chain of amino acids. And so this is the overall process of translation. So this is translation that we see here. We're taking RNA and translating the information in the messenger RNA into some type of protein. So in summary here, protein synthesis is the formation of a protein from DNA. We have two stages, transcription and translation. Transcription takes DNA and turns that into RNA with RNA polymerase. And translation is going to take RNA and turn that into a protein with messenger RNA and transfer RNA. Codons are going to bind to anticodons, and that is going to determine which transfer RNA molecule is going to come in and bind. So this is a summary that I will leave for you all to cover protein synthesis. Feel free to give this a look over. This is just a nice, concise slide. I'll definitely be pulling a couple questions off of this slide, so make sure you give this a look over. So lastly, for Chapter 3, we're going to mention how aging is going to affect cells. Over time, our cells are going to be undergoing lots of different environmental stresses that are going to decrease their efficiency. Their organelles are going to break down over time of all the stress that they might be experiencing, and our organelles are going to lose their integrity. So as we get older, our cells are going to break down, and this is a normal process. This is normal as we get older. We also will decrease the overall number of body cells that we have in our bodies as we get older. So lots of different changes occur as we age, but again, these changes are normal and they happen to all of us. So that will conclude our Chapter 3's presentation. If you have any questions, please reach out to me and I will see you in the next video.